The subject of our discussion was the biosynthesis. Got a weak joint somewhere? The subject of our conversation was the biosynthesis of phenols, and we'd just gotten well started in the shikimic acid pathway, which we'll continue and go on to other pathways today. The shikimic acid pathway we had carried as far as shikimic acid, the key compound, which is this compound here. And shikimic acid is an obligate intermediate and is the key compound. Therefore, it's reasonable that the pathway carry its name. Up until the discovery and elucidation of this pathway, shikimic acid was a very rare compound, just as if you set out to produce pyruvic acid, even though it's a central compound in a number of important biosynthetic reactions, you wouldn't find much of it present. So it was kind of a surprise to find that shikimic acid was as key to these important reactions as it is. The next step that we hadn't discussed is the condensation of a second unit of phosphoenolpyruvate. Another 3-carbon unit is condensed with a condensing enzyme and with the liberation of phosphate, as you would expect, to an intermediate which is shown in your notes but not on the screen. This intermediate has never been isolated, but it would apparently be a required intermediate. But the phosphoenolpyruvate links through the oxygen this time and liberating the phosphate group would form chorismic acid, which has been isolated and identified. Then the chorismic acid goes through a cyclization, which the methylene group sort of leapfrogs up to the carbon where the carboxyl is attached and liberates the oxygen attachment and ends up with the prephenic acid, which is shown both on your notes and on the screen. The prephenic acid has retained the carboxyl group, but all it need do, you see, is to lose this carboxyl group and in the process make the aromatic ring completely aromatic, form the equivalent of the third double bond in this ring. Then the material becomes, in that case, the parahydroxy phenylpyruvate. And the occurrence of phenylpyruvate is no great surprise, which occurs by the loss of water at the same time. So you get two reactions, one forming the alpha keto acid equivalent to phenylalanine, and the other one forming the alpha keto acid equivalent to tyrosine. This alpha keto acid, by transamination, can add ammonia or the amine group rather, and form phenylalanine and tyrosine. So you see the key importance for this pathway is to form those two essential amino acids for protein synthesis. And as we've said several times now, this would then explain the occurrence of phenylalanine and tyrosine in plant proteins, but since animal protein must be derived from aromatic groups formed by plants, then phenylalanine at least must be presented in the diet to the animal, or they can't synthesize protein and therefore die. It is possible for the animal to convert phenylalanine by hydroxylation to tyrosine, so the animal doesn't, or at least most animals, humans included, don't need both phenylalanine and tyrosine, but they do need at least phenylalanine, and certainly will make use of tyrosine if it's available. Now, the two enzymes that are capable of converting the prephenic acid into the alpha keto acid in each case seem to be quite different. Phenylpyruvate aromatase is one, and a dehydrogenase, which could be given a specific name, is the other. So that the nature of the reaction involves a decarboxylation at the position which has become crowded by the addition of the 3-carbon side chain. This carboxyl is easily lost, and that must be lost in both cases. But in the one case, the elements of water are lost to give the phenylalanine analog and retained to give the tyrosine analog, and in both cases forming the aromatic ring. Now this is fairly important again because in plants there are these two pathways, and from phenylalanine or tyrosine on we'll be talking about them slightly separately, or at least the two mechanisms are separate, so that the plant is able to synthesize more or less of phenylalanine or more or less of tyrosine independently. And this comes back to what we were saying earlier, that then in control of this synthesis, the inhibition and repression does relate separately to the two amino acids. And remember, we said the DAHP synthetase seemed to be in two forms, and one was inhibited by phenylalanine, and not only that, its synthesis was repressed if phenylalanine was present, and the other form by tyrosine. So this shows there is a feedback mechanism to control the production of these two amino acids separately in plants, whereas of course in the animal you can't make phenylalanine anyway, so you needn't have that kind of a control. But it does seem to be important in plants, and it's significant, I think, that some of the compounds, the most of the compounds that we'll be talking about now, we haven't really, tyrosine's a phenol, but is fairly incidentally a phenol in some ways, primarily considered an amino acid. Most of what we're talking about then from here on would come through the phenylalanine pathway to the phenols. However, they've been less well investigated from tyrosine, and it is evident that some compounds that retain the 4-OH group of tyrosine might come from this route as well. Now, the two enzymes that go on from phenylalanine or tyrosine to the cinnamic acid, the respective cinnamic acids, and we've only shown on your scheme here the phenylalanine going to cinnamic acid, but tyrosine can go to para-hydroxycoumaric acid or paracoumaric acid, which is the one retaining the OH group in the 4 position, just as well. Now you end up with the trans form of the two cinnamic acids, and the enzyme involved in the case of phenylalanine is shown on your notes as PAL. The PAL stands for phenylalanine ammonia lyase, and that's the name of, or a name of the enzyme, the earliest name of the enzyme, and the one that's caught on. So in the biochemical shorthand, which generally I deplore, but I didn't write it out here, is this is what PAL stands for, to any phenolic chemist anyway, or amino acid chemist. The PAL liberates ammonia, as the name would imply, and forms a double bond to give the cinnamic acid. Tyrase, the same sort of enzyme, does the same kind of reaction with tyrosine, giving para-hydroxycinnamic, but the enzymes are quite separate and rather different in their behavior, and they have little cross-reaction, if any. In most cases, phenylalanine ammonia lyase will ignore tyrosine, and the reverse is a little less strict, but generally is true as well. Notice while we're in this part of the diagram that the arrow from phenylalanine to tyrosine bypassing protein, suggesting that, in other words, phenylalanine and tyrosine go to make protein, but there is a conversion of phenylalanine to tyrosine. Perhaps you might want to write alongside that arrow, plants no, question mark, because that's the way the animals go, but the evidence is that plants don't interconvert phenylalanine to tyrosine and have rather these separate pathways. The utility of the separate pathways is not entirely clear, but at least to me, I don't believe it's clear to any biochemist yet or plant physiologist, but it must have some bearing in helping the plant to regulate the kind of protein it makes and prevent excesses of the different amino acids. Well, the trans-cinnamic acid has no OH group, so that if it's going to be converted to para-hydroxycinnamic, it needs to be hydroxylated, and there is such an enzyme. So even though phenylalanine apparently does not convert to tyrosine in the plant, the cinnamic acid does. So the cinnamic acid can be hydroxylated to para-hydroxycinnamic acid, or paracumaric acid, another name for the same compound, by cinnamic acid 4-hydroxylase, a rather specific enzyme that will carry on this action. While we're talking about the cinnamic acid, the cinnamic acid can be hydroxylated in one of the ortho positions, apparently, and then cyclized, or maybe it occurs as a one-step operation, so that without hydroxylation, you still make the lactone ring. But the coumarin that is formed, and this would be the structure of coumarin itself, is the cyclized lactone or ester formed by linking the cinnamic acid to one of the ortho positions. And it could be either one of the two ortho positions if we pivot around the initial single bond here by the ring. So that if we had hydroxylation, as we will have in some of these other compounds, similar coumarins can be formed, and you can get more than one type depending on which of the two ortho positions one cyclizes to. Coumarins are important compounds, and by and large are phenolic, even though coumarin itself doesn't appear to be phenolic. If you hydrolyze this, say in alkaline solution, you'll break the ester linkage, and of course the OH group that you'll free will be a phenolic OH group. So they're certainly phenol derivatives, and when hydrolyzed become phenols. Note that it's interesting that the cinnamic acid is formed in the trans condition, but obviously it must be converted to the cis condition if you're going to convert it to the coumarin. And this is, interestingly enough, has been shown in the cases where studied at least, to be non-enzymatic. It requires simple irradiation with sunlight, the UV component apparently, and the UV will convert trans and cis bonds, interconvert the trans and cis structures from one into the other. And as a consequence, the coumarin formation wouldn't occur in the dark, at least not unless some cis form had been produced previously before it became dark. So that the coumarin formation would require irradiation in addition to enzyme activity. The coumarins are ordinarily glycosides, and the glycoside sugar would be added after the formation of the coumarin and not, generally speaking, at the cinnamic acid stage, or at least in all cases investigated so far. All right, now if we hydroxylate cinnamic to paracumaric and hydroxylate it again in the 3 position, this becomes caffeic acid. If we methylate that second hydroxy group, it becomes ferulic acid. And if we add a 3rd methoxy group in the 5 position, that becomes synaptic acid, which we have already mentioned the structures of. So this would be the main series of the phenolic cinnamic acids then. The cinnamic acid itself is relatively uncommon and wouldn't be considered, strictly speaking, a phenol, but fits the family. But the paracumaric, caffeic, ferulic, and synaptic are very commonly found, and on your notes you have some examples of interconversions. Now, if tyrosine is hydroxylated as with a second 3-OH group, it becomes what's usually called Dopa, which stands for dioxyphenylalanine, which is German for dihydroxyphenylalanine. And you may know that Dopa is important in certain kinds of browning. Melanin formation occurs in animals via Dopa, so that certainly that mechanism exists, and there are a number of plants that have considerable content of Dopa, the best known is the banana, so that a good deal of the browning of banana skin when it gets overripe, that is, the fruit gets overripe and so on, is due to melanin formation via Dopa. So that occurs in plants certainly, and whether or not Dopa converts to caffeic acid by a similar ammonia lyase type reaction, as is the case with phenylalanine, is pretty much uninvestigated. It appears unlikely, but may occur. Certainly it would give the right compound. And to give you some idea of the other side reactions, we've shown here that ferulic can give the methylene diether type structure, which is listed as methylene caffeic. I don't know that that compound has been identified in nature, certainly not very widespread in any case, but methylene ethers of that type are quite widely found, and this would be the way they're formed by a cross-linking of first the methoxy group on one phenolic group and then that linking to the second OH group in an adjacent position. Coniferal alcohol is produced from ferulic acid, and since ferulic acid comes from caffeic and on up to cinnamic indirectly from them, and coniferal alcohol has the same structure as the ferulic acid, except the carboxy group is reduced to a CH2OH alcohol. Coniferal alcohol is of very key importance because it's the main precursor of lignin. However, synapyl alcohol and the parahydroxy coumaric analogs also enter into lignin under some circumstances. Another example that's given here is eugenol. Eugenol would derive from ferulic acid. You notice it has the same phenol ring structure, the guaiacol ring structure, with the hydrocarbon side chain where the double bond is shifted and the carboxy group is reduced to a methylene. This is important in odorous phenols. That is an best example perhaps of an odorous phenol, important in the oil of cloves odor and eucalyptus leaf odor and a few other places in perfumery and spice odorants. So that some phenols are very important in making the characteristic odor of such compounds. You can call it 4-allyl guaiacol if you like. At least gives you some idea of the interrelationship of the trivial name and perhaps a slightly more systematic name. All right, I think that completes what we want to say about the shikimic acid pathway, except we might reiterate a few noteworthy points as suggested at the bottom of your notes. First of all, note that the hydroxylation pattern, and I hope your notes are corrected, hydroxylation pattern is 4, 3, 4, or 3, 4, 5 from the shikimic acid pathway. So if we have this sort of a structure, whatever the R group is out here, some carbon unit, then we would expect OH substitution in these positions, with this being primary in occurrence and then this plus this being next most common and all three being perhaps least common. And as you'll see later, by and large that's because they're synthesized in that order. The only one that we haven't made very clear here as to where it comes from is gallic acid, and I would refer you back to earlier notes. If you're looking on your handwritten notes, it would be on page 1 of this set. And remember that gallic acid seems to arise directly from 5-dehydroshikimic acid. So that compounds in this series with the 1, 2, and 3, or the phenol, catechol, pyrogallol type substitutions have been accounted for. So that substitution in this 3, 4, 5 type would suggest that the compound arose from the shikimic acid pathway. And this is one of the major points I'm trying to get across in the discussion of the biosynthesis. You can look at the structure in many cases and get a pretty good idea as to why the compound was formed or how it was formed and by what pathway. You can be fooled occasionally, and there are certain compounds that might arise more than one way, and then they need to be investigated. But oftentimes you get a great deal of information by looking at the structure, and vice versa. If you know something about the biosynthetic information, then it would give you a clue as to the structure if you didn't already know it. Secondly, metabolic grid effects do occur, and they make the relationships complex. And substances do originate from more than one immediate precursor. And precursors come from more than one source, so that it is possible for, well, for example, phenylalanine and tyrosine can, in some circumstances, lead to phenols. So, for instance, if you marked a phenylalanine and produced a cinnamic acid, it could make para-hydroxycinnamic, or if you marked tyrosine, it would also. So you could have dilution of marker if you only made one radioactive, or you could have different ratios if you made both, or different fractions of both radioactive. Another point to be made, I think, is that while we're focusing on the shikimic acid pathway as a source of phenols, the early and perhaps more important part of the structure from an essential metabolic viewpoint is to produce the amino acids that are essential for protein production. So this means that the production of phenols other than tyrosine would be taking carbon away, if you like to put it that way, from an essential metabolic pathway. And we'll come back to why that might be important later on. Not only that, then you see that pyruvic acid appears into the formation of these compounds. Well, that indicates carbohydrate metabolism, or possibly fat metabolism, depending on where it comes in from, and would indicate also with the erythros an important relationship to metabolism. Since the pyruvate is probably produced either by respiration or anaerobic Embden-Meyerhof type carbohydrate breakdown, then one would expect the Embden-Meyerhof scheme to have some relationship to how fast phenols are synthesized. But vice versa, since the erythros unit is produced by the hexose monophosphate shunt, then the ratio of metabolism of carbohydrate between the two major pathways of carbohydrate breakdown would be significant. And it turns out, since pyruvic acid is generally present in what appears to be metabolic excess in the sense of these reactions, if one wants to favor the synthesis of phenols by this mechanism, then shifting away from the Embden-Meyerhof type sugar metabolism toward the hexose monophosphate type, or the pentose path, if you like, does favor the synthesis of extra phenols. And again, we'll come back to some suggestions why that may have some significances in environmental response and that kind of thing. I think that covers the main points that we wanted to bring up about the shikimic acid pathway. Now let's consider the conversion of phenols that don't arise by the shikimic acid pathway. The next most common, or perhaps even in some ways more important pathway for phenol synthesis, is the conversion from acetyl-CoA or malonyl-CoA in the fatty acid or fat type metabolic scheme. This is outlined again in your notes or also on the screen. You start with an acyl starter group, which we've shown as the equivalent of any R group, with the acid function, carboxy acid function, converted to the thioether with the sulfur group of coenzyme A, which we won't go into the structure, but it's a pantothenic acid-derived substance that I am sure you've had in biochemistry. And the main point we want to make is that the thioether in this case is essentially a high-energy bond that then enables activation and reaction in other ways. Now, actually, we old-timers learned this largely as the acetate derivatives, but by and large, the reaction of the kind we're talking about comes from malonic acid rather than acetate. And we've shown the reaction, both in your notes and on the screen here, as the R acyl starter group linking with a malonyl-CoA unit, which is basically the same. It's like one group linking with another, you see. And what happens is the electrons of this bond would link with the carbon that has the carbonyl, and the thioether bond would facilitate, this high-energy bond would facilitate that reaction. Net effect would be to liberate this carboxyl as CO2, and the one unit of CoA as the SH compound, with the energy being used to form the new bond. So you would be forming what might be called a head-to-tail type linkage, in that the carboxy group is visualized as the tail, and this was originally a carboxy group, and the methylene group, which arises from here, would be, as from an acetate sense at least, would be the head of the compound, or the less reactive part. So you'd get head-to-tail linkage, and the net result is that if you repeated this a number of times, and as shown, this would be the acyl starter, and then this would be 1 malonyl, 2 malonyl, and 3 malonyl. Did I lose one carbon here? No, here I drew it too far. Okay, this is the acyl starter. That's the 2 carbons from the first malonyl, the 2 carbons from the second, and the 2 carbons from the third malonyl-CoA unit. So that as drawn here, then, the net result would be the polymerization of an acyl starter, which might be another acetate 2-carbon unit, or it might be something else. We'll come to what something else it might be shortly. And then the 3 acetate groups. Now you notice that, or the equivalent of what forms the 3 acetate type 2-carbon units. If we number these in the order shown, 1, 2, 3, 4, 5, 6, 7, then we can link this, we can cyclize this in more than one way. And apparently it appears that the enzyme that does this linking holds this multiple beta carbonyl compound on the surface of the enzyme, and then as it releases it, or in the process of escaping, the compound is held in certain ways so that it can cyclize in more than one way, but it is specifically controlled as to what cyclization occurs. So it does appear that the linking, something like, well, we believe now that proteins are linked together by sort of stitching on the amino acids in units, and then letting the tail of the protein escape out into the surroundings of the enzyme, so that it's held in place until it's made, you might say, at least from one end. In this case, we think it may well be held sort of spread out on the surface. In any case, you don't find the poly-beta keto compound free in the solution if you make an enzyme preparation capable of doing this kind of reaction. And I don't believe you can completely free of particulate matter, but if you have the mitochondria and the proper particulate matter, it can be done in the test tube. But you don't ever find the poly-beta keto group free. These phenols formed by, and other compounds formed by this mechanism, are sometimes called polyketides, in the sense that they do have this polyketone type structure. Now, while we're on that point, it does appear that there is some finite limit to this enzyme surface. A common group of phenols, and you'll see why they're common shortly, are the groups that have formed from 3 acetate equivalents or 3 malonyl-CoA equivalents and a starter group. But it does appear that enzymes are capable of taking up to about 10 acetate equivalents, and then the molecule apparently either becomes so perhaps water insoluble or too big to fit on the surface of the enzyme, or some such reaction. In any case, about 10 acetate units seems to be the limit of this kind of biosynthesis. Now let's talk about the cyclizations. The cyclization, which might be called carbon acylation, and by carbon acylation we mean if we have a structure where there is the carbon on one end linking to the carbonyl, then carbon acylation means a covalent bond, single bond, between a carbon and the carbonyl carbon. So that's, whereas O-acylation, we'll talk about shortly, would be forming an ester group, you see. So O-acylation would be forming an ester type linkage with the carbonyl, and the carbon acylation would be forming a keto cyclic, or keto, in this case, cyclic structure. All right, if we have 1,6, so if carbon 1, and this, of course, as assembled on the enzyme, would still have the acetyl-CoA, or the CoA thioether unit still attached, probably. That might facilitate cyclization. In any case, we can cyclize the 1 to the 6, and this carbon that will be connected to the acyl group then would be the 6th carbon, and the acyl group, or the carbonyl, would be provided by the number 1 carbon. If we do that, and you may want to play around with this with pencil and paper, if we do that, we get this kind of structure, which would have the R group and the acyl group beside it still as they were, then the 6th carbon starting the ring, and that would make a 6-membered ring, and it would have the general fluoroglucinol type meta-hydroxy substitution. And notice that if you make the cycled compound, what you have done is you've included 3 carbonyl groups in the ring, or attached to the ring. So if we have this structure, did get the OHs in the right positions, yes. If we have this structure, then this would go immediately, it's tautomeric, but there'd be very little tendency to remain in that structure. It would go immediately to the enolic arrangement, where we have the OH groups. So we don't have to remove any hydrogens or any other type of reaction. Once formed, a triketo cycle like this is going to be immediately enolized to the fluoroglucinol type derivative. Now, if the R group was a cinnamic acid derivative, so that if we had something like caffeic acid and in this acyl starter group, then caffeic acid would lead to a chalcone at this stage, and then the chalcone, by reactions we'll show shortly, would convert directly to a flavonoid. So you see why this is perhaps the most interesting and most important type cyclization, therefore shown first, in that this would be the way that you would form flavonoids, starting with a cinnamate. Now we're talking about what we've described as the acetate biosynthesis pathway, and the acetate results in the fluoroglucinol type substitution, which is quite visibly different from the 3,4,5 type substitution, in that it would be the 1,3,5 meta type of substitution. But in the case of flavonoids, you see, the R group would have arisen from the shikimic acid pathway, and then what is going to become the A ring would have been synthesized from the acetate pathway. So in fact, both pathways are involved in the synthesis of flavonoids. Now we can link this in several other ways, and in each case we have natural products that are produced, or at least their structures would indicate. They haven't always been proven that they were produced this way, but their structures would indicate they have been. And one type of linkage mentioned is the 2,7 aldol type condensation. All right, if you link carbon 2 and carbon 7 in an aldol type reaction, and you may want to look up what aldol reaction is if you've forgotten, you'll get this sort of substitution. This OH group then is often lost by dehydration, and you'll notice if that happens, you'll generate a double bond here and then make the aromatic ring. So that fungal products often arise by this mechanism. Many products of molds and lichens have structures that arise this way. And an example is given on your notes of orsolinic acid, which would be a direct derivation without the participation of anything other than acetate. But the dehydration would lose one OH group, but the remaining OH groups in a resorcinol type structure are still meta. So this would be one example of this kind of formation. And Pinal Sylvan is another compound which we've mentioned, but I don't believe the structure. Yes, I guess I drew the structure in on your notes. So Pinal Sylvan, as you might guess from the name, comes from pine heartwood, and Pinal Sylvan is a still bean, and the still beans apparently arise from this kind of reaction, but in this case with a cinnamate side chain participating, and it becomes the R group. In this case, notice that the compound has been also decarboxylated, so that this CO carboxyl group has been lost, so that more than one reaction may occur in modification of the structure after it forms. But if you trace this back through radioactive markers and so on, it is shown that the acetate type pathway was used to form the first ring, the top ring as drawn on your structure. 1,5-O-acylation, which is formation of a lactone then, does also occur. So carbon 1 can link with carbon 5 after we visualize carbon 5 is enolized to an OH group. So if it's an OH group on carbon 5 and an acid group on 1, then the lactone between the two can be formed. Again, if you trace back the keto groups, you'll see that the keto groups are as expected, except an OH group that would be indicated in the position here may often be lost. And one of the most obvious examples of this kind of linking is cavaine, a constituent of kava. And if you've ever heard of kava, if you've ever been to the South Pacific, you've heard of it. It's a narcotic or a drug drink. You may have heard of the kava ceremony from the Fiji Islands and so on. It's a very interesting substance, a plant. They take the root, mash it up, usually get the prettiest girls in town to chew it up and spit it into a bowl, and then after a certain amount of fermentation, the people drink it. Of course, it'd be nice to have the prettiest girls doing it because, well, it doesn't sound all that attractive. It turns out that there is an enzyme in saliva that breaks down some of the material, therefore it's necessary to chew it up. You can't get the same effect if you just pound it up. In any case, this narcotic produces a euphoric state, a high if you like, and it seems to be a very interesting one. Apparently the Polynesians have just about killed all the kava in the Polynesian area. It's very hard to find the plant anymore, and so even though I lived in Hawaii a good length of time, I never saw one, never got to taste it. But they say that this has a very nice effect in that it makes the world seem bright and shiny and pleasant, but it paralyzes your nether extremities. So you have to sit in the corner and smile, and you can't get into any trouble, you see. So it's a pretty good drink in the sense that it keeps you from wandering around and getting into any trouble. And the active ingredients include phenolic derivatives like cavaine. There are others as well. Some alkaloid apparently is present, but it seems that the phenolic derivatives are the important ones in producing the effect. And while we're on the point, I might digress a little further. There is a characteristic dermatitis produced in cavaholics, if there are such people. Anyway, people that consume this a great deal, when they get in the sun, get a characteristic and very severe dermatitis. And there is a light reaction then to the phenol in the body, and we'll have occasion to mention other examples of light reactions where if you consume certain plant phenols, it makes your skin very sensitive. I think we did mention the hypericin in the case of cattle, where a white and black spotted cow will get a terrible dermatitis in the white spots only if they eat St. John's wort, take in hypericin, and then stay in the sun. So this is a fairly esoteric example, but also a rather intriguing example of a natural product that has a lot of physiological activity in animals, and what benefit or reaction it has in the particular plant we don't know, but it's very limited distribution in a certain few plants. Well, the next type of link, or the remaining kind of link, is an acetal or ether, depending on how you look at it, 3,7 oxygen linking. So if you link between carbons 3 and 7, both of which are carbonyl bearing and form an oxygen link, and I find it easier to think of it as an acetal. So if the first link is between one of these enolized, and as drawn it would be the 3 carbon, if you visualize the 3 carbon as enolizing and then linking with the, no, I take it back. The 3 carbon would be reacting in this case as the carbonyl, and the 7 would be enolizing and reacting as the OH group. And you know the reaction between a keto group and an alcohol group to give an acetal. And in this case, we'd visualize this as a hemiacetal if we take that kind of approach. Now the OH group is readily lost, and then you would end up with chromones of the structures shown here. And in fact, as you'll see shortly, this is basically the reaction to close the heterocyclic ring on the flavonoids. So it turns out that in flavonoids we have both kinds of linkage. We have the 1,6 carbon acylation to form the chalcone type structure and the fluoroglucinol A ring, and then we have the 3,7-O acetal type formation to form the heterocyclic ring. And it might even illustrate why the flavones were considered the type compounds, because if this had a phenolic group in the carbon adjacent to the oxygen, then that would become a flavone. So these are the four general ways that this acetate polymer can cyclize and give other phenols. Noteworthy points in this regard are listed at the bottom of the page. First of all, has been clear from the beginning, the OH substitution would be meta, and this in equivalence then would be one acetate type unit, where this would be the carbonyl of the original acetate, and this would be the methyl group of the original acetate. So we could, and this has been shown by marked acetate, if we follow these back, then this methyl group produces this, and this carbons in the ring, and the original carbonyl produces those that have the OH groups attached. So that the meta arrangement with a carbon in between, OH groups with carbon methylene groups in between, would be the characteristic structure of this kind of synthesis. We've also shown that the two shikimate and acetate pathways cooperate to give the flavonoids. We've mentioned that this polymerized structure seems to be held on the enzyme, and presumably one reason it's held on the enzyme is that the enolic form of the beta keto groups form strong hydrogen bonds, and so presumably this helps bond the polymeric unit to the surface of the enzyme. And we've indicated that, counting the starter group, 4 acyl groups are most often linked, in that the flavonoids are the most important compound series produced this way, but up to about 10 acetate units appear to be possible. So if you had 10 acetate units, that would be 20 carbons. So a C20 unit might be reasonable. And also, you would expect this to give in enology, and remember the acetate path is tied into fat synthesis. So now we tied the other one into amino acid and carbohydrate breakdown and synthesis. And in this case, we're tying it into lipid synthesis. So you see all the major pathways then are converging, if, to use my terminology, converging in formation of phenols. Diverging might be the preferred term by a lipid chemist or someone else. Since we're linking the equivalent of acetate units, you'd expect to get even-numbered materials. However, it's easy to decarboxylate a number of them, and so then you could arrive at odd-numbered derivatives. So, and examples of the decarboxylation have already been given, like in the case where this carboxyl was lost after cyclization. So that ordinarily it's not as clear-cut. In the fatty acid series, you can generally count on it being an even-numbered carbon series acid, unless something rather drastic has happened. And apparently the reason for that is that in fatty acid synthesis, you don't retain the carbonyl groups as you form the polymer. In other words, you link the acetate groups, and then you'd be reducing these as you go along, so that you don't have the opportunity to snip off something along the way and end up with an odd-numbered carbon. Whereas in our case, we do retain the beta-carbonyl structure, apparently, or the beta-enolic structure, until we begin cyclizing the compound, and that beta diketone type units, or beta keto acids, are very subject to decarboxylation. Another point is that the methylene groups are activated in the sense that they're made relatively negative, so that the methylene groups become reactive, and it's possible then to make these cyclized type links and perhaps other linkages that would result in some of the structures that we do see in nature. addition of a methyl group or an isopentenyl group or other hydroxyl groups, which are electrophilic, is possible, evidently, at these electron-rich or relative negative methylene groups. These secondary effects, such as hydrogenation to form alcohols, you see if you have an enol, and you reduce the double bond, then you've stabilized a secondary alcohol, and it would be rather difficult to reverse the reaction. Then dehydration of such a structure would give a new double bond, but without the OH substitution. So that a number of possible reactions secondary to the acetate type chain formation do occur and can account for a number of these other results. Now, in just a few minutes remaining, I'd like to talk about a third pathway, which is somewhat less important by and large for plant materials. Less important in the sense that there aren't so many compounds made this way, although since some of them are quite important, such as the ubiquinones and so forth, then it might not be fair to call it lesser important. And apparently it's the only way that animals can make phenols directly. Again, starting with 3 units of acetyl-CoA, and apparently in this case it is acetyl-CoA and not malonyl-CoA, although it's possible to produce acetyl-CoA easily enough from malonyl-CoA. 3 units linked together in a new way to produce, as an early reaction, the beta-hydroxy beta-methyl glutaryl-CoA, which is shown first on your notes and on this screen, and then mevalonic acid, or the salt mevalonate, and this is a branch chain unit with one of the methyl groups. There's a total of 5 carbons, and one of the methyl groups in the middle is in a side chain. Then with the addition of energy from ATP, this is converted to mevalonate 5-pyrophosphate and decarboxylated, so that you end up with, no, excuse me, it was 6 carbons before. When you decarboxylate, you end up with the isopentenyl pyrophosphate, which is the 5-carbon unit. And there are 2 forms of this, depending on whether the double bond is on the 3 or methylenic position, or the central position in the molecule. So you have 2 forms of isopentenyl pyrophosphate, and these are C5 units, and the 2 forms are in equilibrium with each other. When 2 of them link together, you get a terpenoid, such as geranyl pyrophosphate, which, since you're linking 2 5-carbon groups together, would be a C10. So it's C10 unit of the geranyl pyrophosphate form would be produced. And if that is oxidized to linalool, and this occurs by biochemical methods we won't try to delineate, but in any case, linalool, as you may know, is the odorant of significance apparently in muscat wines and grapes, so that this would illustrate how this kind of compound arises. If the C10 group adds another isoprene unit, and we'll come back to what isoprene is in just a minute, another isoprene unit, so you have a total of 15 carbons, then you'd have farnesyl pyrophosphate, and 2 of these linked together would give a C30 compound, and these would be keratines, so that keratines are synthesized by this pathway. Well, up to now we haven't had any phenols, but if we cross-link with the pyrophosphate unit to make the cyclic compound and dehydrogenate to make it aromatic, we have thymol, which is again a constituent of cloves, and thyme, which is a mint family product and does have aromatic character and has other interesting effects. For instance, thymol has often been used as an inexpensive antiseptic. So if you're doing Warburg respirometry, say, and you want to make sure bacteria don't grow in your system, then you can put in a small amount of thymol, and usually it'll inhibit bacterial growth and enable the enzymes to go ahead and work. Well, it looks like I'm just about out of time, so we'll continue what little more we want to say about the isoprene path and interrelationships from here on next time. If you have any questions at this stage, it's